All right. Welcome to the Crescent Method. This is the Science of Living Soil Systems, Part 3. If you've not watched Part 1 and Part 2, we will link those in the description below. And I suggest you watching Part 1 and Part 2 before you check out Part 3. For those that are up to speed, here we go. I've spoken about myself quite a bit, and one of the things that I don't talk about is some other things that we used to whoop ass on as a kid. Growing up, I come from a racing family, and we took it extremely serious. We would take a tremendous amount of data sets, and even in 4th, 5th, 6th grade racing go-karts, we would take a tremendous amount of data. So we would take inside, middle, and outside tire temperatures. We would measure stagger, which is the size of the outside diameter of the tire. We would take weights, cross weights. I mean, we were doing full NASCAR shit when we were in 4th, 5th, 6th grade. And that led to track championships. And one of my jobs, because I was the youngest, was when we moved up into the stock car age, when we could race actual cars, my job was to reach under the car after the car came back in after a lap and take, all the tire temperatures, air pressures, and then I would also log track times. And so at the end of each race and at the end of each practice session, we would compile these massive data sets and we would chart it out. And we were taking things extremely seriously. And like I said, we won a lot of track championships. And I learned even in 5th, 6th, 7th grade that if you're going to win, you're going to get haters. And luckily, my family coached me on that as a young man, that to be at the top of the pile, there's going to be some crabs. And to just keep doing what's good to you and keep being true to yourself and be grounded in the results that you can prove to yourself with taking the appropriate data sets. And so from a very young age, we were compiling massive data sets on everything that we would do, and we would turn that into actionable advice, and we would apply it to the things that we did. This was from sports to racing to everything else. And when we got into regulated cannabis, it was no different. As soon as I got in there, we started taking data sets, a tremendous amount of data sets, actually. And once Sarah and I got together and started doing the microscope methods by Dr. Elaine, we took the same approach moving into that. And when we would show up to a cannabis facility, we would do a tremendous amount of microscope data so that we could provide real-time vital statistics to the cultivator so that we could very confidently move forward in our steps to make action based on the data sets that we were taking.
Yeah, so many of you know, and if you don't know, I was a nurse for 10 years. And so a lot of my background is very similar to Scott's background in that, Western medicine, we do a lot of data. We take all the vital signs, we get lab work, and that helps us determine what's going on with the patient, what next moves that are appropriate to take that should improve the patient's life. And the patient is the ultimate determiner whether or not that is that actually improved their life. But we need these vital signs and we need the stats in order to make educated decisions about what those next steps would be. So that's the mindset that I come in with, is what are those data sets that I can learn, utilize, and actually implement that make those changes in the plant, that make the plant thrive. Because if that's not the end goal, and if that's not what the response is with the plant, then what are we doing? So you start to look at what those data sets are. Does it make sense? Does it shake out? Does this theory actually prove itself time and time again? So that's a bit about me and how I think and how we think together about why we do what we do and how we put together the tools and scientific knowledge that we have learned over the last 6 years doing this work. So what are some of the facility considerations that we need to think about when we intend to utilize things like the soil food web or any organism that we want to put out, right? So you want to make sure that you are distributing those organisms to every plant. We certainly intend to do that. On the variation of those microbial populations, it can be very wide. You can have lots of variation in those populations that make for inconsistent use of these nutrients and the water that your plants will take up. We intend to distribute them evenly. We intend to make sure that there's no variations in those things, but we do see that happens, and what leads to that. So that's what we're going to look at in this particular video. We're going to look at what those variations are, and we're going to just take one particular organism and track that very easily to identify for yourself. And then how do these variations in these microbial populations actually play out as it pertains to pest problems? So as those pest problems pertain to a greenhouse environment, the location of your plant, what that plant is experiencing, any hostility in the environment in that location, to a wet wall or a draft or extra humidity, those kinds of things. So we're just going to track, as you see as we go along here, one organism, and you'll see how we do that. So let's kind of get down to some ideas about the soil food web first. About, we intend to utilize the soil food web. And so, if we think that we utilize the soil food web, then maybe we get the soil food web, right? If we intend it, we get it. If we want a diverse soil food web in our garden, then we should get a diverse soil food web in our garden. If I'm distributing these organisms, then I'm evenly distributing them, regardless of my process and my materials that I'm using. And then, we can often think that our growing room is exactly the same from corner to corner. Our greenhouse is exactly the same, that there's no variation. So those are some of the ideas that we can hold sometimes that may not actually serve us in basically solving problems and getting to the root of what's happening and why we're getting the results that we're getting.
Yeah, people often don't understand that there's a bad corner in the greenhouse, and it's because there's a variation in wild microclimates, or there's a variation in environment from corner to corner. And all these effects have dramatic impacts on the soil food web and ultimately on how your facility performs, regardless of how big it is.
And so, we throw this term around. What does soil food web mean? Generally, it means the soil food web. So there's a food web. We're all familiar that there are base organisms that have higher organisms that eat them, predator organisms, and so on and so forth on down the line. And that's the same for in the soil. These are those microbial populations where you have a base organism set, and then you have the next microorganisms that eat them, and then the next ones and the next ones. And those are called trophic levels when you get down to the scientific terminology. And there's a thought, and the word I'm looking for. There's basically a thought called ecological succession, and what that is that, basically speaking, your plant has evolved with a certain set of microbial populations in all of the microbial organism groups. So any particular plant will have evolved to see a certain set, a certain biological quantity of each of these different organism sets. And so from that, we make a term to talk about that called biologically complete. You'll hear Dr. Elaine say biocomplete, and that's what that means. It means that for that particular plant, it evolved with this microbial profile, this set minimum of organisms that it needs in order to have full function and full health and vitality in that particular plant. And so in order to achieve that biologically complete set of organisms, we have to quantify it, or we don't really know what's going on. And so soil food web to us that are trained in soil food web means that we quantify it, that we actively look for all the players on the field Do we have them? Are they in the right quantity? Is there one that's higher than another? And do we need to bring that down? What do we need to adjust? So we want all of those microbial organism groups and present in an appropriate ratios for your plant. And so we encourage you to have your soil quantified and then look at your practices and start to, if you can remove the charge, the emotional charge from what you want to happen and allow what is happening to show you what you could adjust and what that means. So like if you've got too much bacteria or you've got too many oomycetes or a lot of ciliates, what does that mean for you? And how would you adjust that? If you start looking at it, you quantify it, you can know whether or not you're watering too much. You can know whether or not you're adding too many nutrients. And it's just kind of having that courage to do that and to adjust accordingly. So, and we would say that soil food web is not just using fermented bacteria or mycospores or bottled bacteria or IMOs. It's not any one of those things, and it's not even all of those things. Those, I would say, would be about 1/1000th at best of the entire organism populations that you should be having in your soil. So though it is better than sterility for sure, it misses the mark on what could be. So in our, when it comes to soil food web, we want all of them. We want that redundancy. We want many bacteria there present doing the same jobs. We want redundancy. Diversity of organisms leads to balance and leads to stability and high health and yield.
So we're going to now walk you through a real life example from our consulting work. This is a project from 2017. This was a roughly 53,000 square foot greenhouse. It was, a Dutch house, so it's 5,000 square meters, as they say. But essentially, there was 16 pretty identical bays. They were roughly 3,200 square feet. And each bay that had moms in it would have roughly 150 30-gallon pots, or it would have something like 120 65-gallon pots. This particular facility was testing out some different strategies, and at the start, it's hard to have enough soil to really fill up beds, so they were tinkering with 30-gallon pots. And so when you talk about 150 times, man, there's probably 12 bays filled up. That is a tremendous number of ecosystems to manage, and that's where Sarah and I come in, is we quantify those ecosystems. We put very specific metrics to it based on the Dr. Elaine Ingham soil food web analytic strategy, which I still stand by is the most accurate way to assess the agricultural process. And so we implement that in our professional work. So in this situation, we wanted to track the health. We wanted to quantify where the vulnerabilities were. And for this particular example, we're going to track just one single organism, because the entire microscope qualitative assessment is extremely complex, and it's really hard for people to wrap their mind around the complexity of it. And so we're going to look at one single organism, which is a nematode. In this particular example, we're going to talk about nematodes that were predatory. This is a bacterial feeding nematode, and we know because it has the mouth parts here for sucking up bacteria, and then it has the bulbs here for squishing up the bacteria, the median and terminal bulb, and then these are actually the bacteria inside the stomach. And so what you're looking at is the 400 times magnification, or if you have a microscope, that's usually the 40x with the 10x eyepiece objectives. And the 400x is where we do the very specific analysis. And if you know what you're looking at, you can very, very clearly tell the difference between bacteria and other sediment and rocks. And so Dr. Elaine teaches very specifically how to quantify these organisms based on her 4 decades of research doing this technique. And we wanted to use this example for this episode because this is something that you can quantify yourself. From the point that I became qualified to perform the qualitative analysis from Dr. Elaine, it took me like 1.5 years before I felt that I was accurate from the day I learned the basic principles. And But this is something, if you own a microscope, you can do today. If you've been using nematodes, if you believe or hope that there's nematodes in your compost, this is something that you can do today. And so what you want to do is, if you have a test tube, you want to put 1 ml of compost into that test tube, and then you want to add clean, non-chlorinated water up to the 5 ml line. If you don't have a test tube, just make a basic 1 part soil, 4 parts water, and gently shake it. We do this little thing like that, and we do it 30 times very gently. And then we take an eyedropper pipette and we pull out 1 drop. We put it onto our slide. We put our cover slip on there. And then you move to the 10x objective, which would make 100 times magnification. And you literally go from the top corner and you work your way down, move over a field of view, go all the way back up to the top of the slide, move over a field of view, go all the way back down. And we call it a nematode scan. If you get good at it, it takes 20, 30 minutes. When I first started, it did take like 45 minutes to an hour to perform this entire scan. But if you own a microscope, I would encourage you to do this today. I know to the average person, some of the claims we make about what we can do with the microscope seem pretty wild and far out. But this is something that everyone can do to see if what we're talking about has any validity. And so in this particular situation, this farm was using a 55-gallon barrel to dispense the compost extracts. They would then add the predatory nematodes to the barrel. And because this barrel is flat bottom, all the nematodes are going to sink to the bottom. And nematodes have a density greater than water, so they actually sink quite quickly. Within a matter of 10 to 20 seconds from when you put them in this tank, they're going to be at the very bottom. And then they were using a 1/6 horsepower transfer pump to dispense that through a garden hose, which is actually our preferred method. This is something that we taught to this facility, and they were doing a pretty decent job at distributing the organisms. But with any scale, there's always stratification, there's always variation, and you have to try and manage that. And so this is meant to be an overhead view of a single bay. Obviously, it's not to scale. It doesn't have 150 pots in it. But nonetheless, the facility had 5 rows of plants. And what they would do is they would start down here with their applications, and they would put their tank right about here. And then they would fill this entire row, and they would dispense. Then they would bring their hose back, and then they would go down this row, and they would dispense to all the plants. And they would finally get this last row. And because they were consistent from bay to bay, when we looked at this when we looked at this with the microscope, we found that the nematodes were not being evenly distributed. And what ends up happening, because it's a bottom tank or a flat bottom tank, oops, sorry, it's my first day drawing. All the nematodes go into the first, basically it was like the first 10, 15 pots, I think. And because they're dispensing this way, they go to these first pots right here. And almost no organisms got to there, almost no nematodes rather. And so when we went through this space, because of we know they were starting here and they were working this direction, that gives us an indication of how to analyze this property. So we took a representative sample from here, we took a representative sample from here, and we take a representative sample from here. And we analyze all those. And when we looked for nematodes, we saw that there was almost no nematodes in most of this region, actually. And when you scale that up to 16 bays of 53,000 square feet, this is the amount of facility that was actually protected from pests, from those applications of nematodes. This is the amount of facility that was vulnerable and was not receiving any protection from pests from those predatory nematodes.
So the intention was there. They thought they were doing it.
Right.
But come to find out, there was a learning lesson to be had about this particular organism and certain qualities it had that lend itself to this pattern.
And this phenomenon happens everywhere. Whether you're running 6 pots, 2 beds, or 4 acres, the same phenomenon is true. And we do find with nematodes specifically, there's a tremendous problem in the industry with how people dispense these based on the advice that's given from pest people. And when we looked at the facility, this is all of the samples, all of the distribution, And you can see quite clearly there's a significant problem. This is another project that was in the same year this was actually 2016. And this was a large 20,000 square foot facility. It had 4 5,000 square foot bays. And in each of the 5,000 square foot bays, they had a tank like this. They call it a horizontal leg tank. They're quite affordable. They don't fall over. They're easy to reach into. When you get into the commercial space, there's a lot of OSHA violations. And so you need things that don't involve ladders. So the positive aspects of this tank was that it didn't require a ladder. Almost any employee could reach into it, and it could hold a large volume of water. And then they had a valve down at the bottom with a bulkhead so that everything was being distributed from the bottom. And so what's the same about the context of this facility is that it was a flat bottom tank. The nematodes were being put into the top of the tank. The nematodes have a density greater than water, and they sink to the bottom almost immediately, and they get stuck in all these little nook and crannies. And so if your tank has a bulkhead down at the bottom, it's not picking up the bottom 2 inches. So most of the nematodes that are put into this tank actually die at the bottom. And this was actually the advice of an IPM manager, and it shows that they don't use a microscope to quantify their process with respect to biological organisms.
One more thing about that tank too is they did actually have a little bit of aeration, so they thought that they were getting good even mixture, and anybody would think that as well. So it was news to them that this was actually not sufficient enough for what they were intending to happen.
Yeah, what people don't understand is that organisms are so small in relation to an air bubble, and those air bubbles don't come from the very bottom. That's why cone bottom tanks are important, because by default, it funnels everything down to the bottom to be blown back up with the air. And so if you have a flat bottom tank, you can aerate the shit out of it. And because of cavitation, those organisms are bouncing off the top of the air bubbles, and they literally just fall to the bottom and then die on the bottom. And so aerating from a flat bottom tank does almost nothing for homogenization of organisms. And so this is meant to be a overhead view of the 20,000 square foot bays. Down here at this X is where their tanks were located in the center, pretty reasonable there. They had a hose reel that was right in the center, pretty reasonable. And then because we like to get high and do our job, we like to do it the exact same way every single time so that we don't screw it up. So employees would come in, they would stretch out the entire hose, and they would go and they would do this zone first. Since the hose was stretched out completely, they then would take it to the other side and do this side. They would reel up the hose a little bit. They would hit this zone second. They would hit this zone third. They would coil up the hose a little bit more, and then they would finally hit this location second. And so what do y'all think would happen last? Yeah, so what do y'all think would happen with the nematodes, and where are they going to go? And when we show up to this facility and we perform a microscope analysis based on their explanation of how they're behaving, we know how to sample. In this particular facility, we did a horrendous amount of soil sample data while being there through a week, and painted a remarkable picture about every aspect of the behavior of this facility, where the vulnerabilities were, and where the opportunities for success would lie. But for the sake of simplicity, we're going to talk simply about nematodes, and so because they're going here first, that's where all the nematodes go, 51 nematodes per cover slip. That is a king's ransom of nematodes. They would come here second, they would come here third, they would come here fourth, and they would come here fifth. And this pattern was repeated through the entire facility. And so this particular group right here was the oldest in the bunch. And what they would do is, and why this was so confusing to the farm, is they would fill up 2 rows at a time week 1. They would fill up 2 more rows week 2, and then they would do a single batch on the 3rd week, and then they would move to the next greenhouse. They would fill out this group, fill out these 2, and then fill out this one. And they kept repeating that pattern all the way around in a circle to here. And you can see very clearly the pattern is identical until you get to here. And you must ask, well, why is the pattern different here? Well, this is the week they ran out of nematodes. So this group had been given nematodes, and they started here with 2 groups of plants. So I guess they were going that way. They did 2 groups, and 1 group. But nonetheless, they were watered in the same exact pattern. These plants got filled up in the weeks after, but they had a 2 or 3 week lapse where they didn't have any nematodes. And this facility, they didn't bring us for problems. They brought us here because they couldn't figure out why the end bays, despite being part of the same group, but because of this massive discrepancy in nematode populations, there was a very measurable impact on yield from metric. And they could not figure out why the middle groups in every single bay, regardless of when it was brought into the building, was consistently underperforming, was consistently higher in pest damage, and there was just correlated problems as well.
Okay, so what we suggest actually to solve this problem as a low budget strategy, in lieu of, the fancy dram buckets and these flat bottom tanks with some sort of aeration, is to literally, when it comes to nematodes, just take it into your own hands, get yourself one of these scientific wash bottles, basically, and very inexpensive, and then get yourself the nematodes. We definitely advocate sound horticulture. We do not get paid by sound horticulture. We would be rich if we did. But in all of the last 6 years of testing nematodes and trying to find which company that we should really give our service to, sound horticulture, hands down, always has the quantity that they say they have, and they're all alive. I've never received any nematodes from sound horticulture that has not been alive. Not one. And
accurate too. I think only maybe one time we saw the Steiner Nema and the HBs mixed up. They're accurate, and the populations are true. You know.
Yeah, so definitely if you use no other nematode, you do want to use the HB nematodes, the Heterorhabditis bacteriophora. Those are bacterial feeding, and they harness a certain bacteria that does the work for you for the pest control, and that's what actually takes out the larva. So those will do twofold for you. They'll cycle nutrients in the soil food web, and then they'll also, because of the bacteria they harness, they'll affect those larvae that you're after. And then the other two that you can add in addition to those, to the HB nematodes, would be the Steiner Nema carpicosae and the Steiner Nema feltiae. All three of those, or just the HBs, are good. But when you use these nematodes, we suggest that you use a small volume of water to extract them, put them into this wash bottle, and you can see that literally the wash bottle does suck up from the bottom. So everything that we've just been talking about needs to come into play here. So what do? We just tip up the bottle back and forth so that it's mixed. Squirt one or two squirts at the base, in the rhizosphere of each plant, and then, go back and do it again. Between each plant, you'll want to mix that up so that you're not pulling all the nematodes that have settled at the bottom, so that it's mixed and every plant actually gets the nematodes that you intend to do. You paid good money for this. You're putting all this effort into it. This is how you can do it accurately and inexpensively.
So this was another interesting example that takes into consideration the full soil food web. And so this was a friend of mine that had 2 large beds, and he had a tea brewer that was a cone bottom, so that's beneficial. The problem he had was that down below, he only had the way to hook up the air to the valve, but when he wanted to dispense, he had to turn the valve off, disconnect the hose for the air, connect the hose for dispensing. And in just the time it took for that to happen, you get tremendous stratification of the organisms, and all the nematodes obviously go to the bottom that we've just spoke about. But the other organisms as well, they start to sink. And just in the amount of time it took him to hook up the hose was enough time to create remarkable variations. And because he likes to get high and do his job and not forget, he does it exactly the same. And so he would dispense to bed 1 first, and then he would dispense to bed 2 second. And after his second harvest of very clear differentiation in yield, outcome, pest pressure between bed 1 and bed 2, bed 1 was consistently and remarkably outperforming bed 2. And when we look at
And that's when he reached out to you. He's like, what is going on? Take a look at this under your microscope. Tell me what's going on.
He was asking me what was up. And, I told him, send me a sample from each bed. And this is what we have.
Okay, so this is just the bioassay. And I break this up into 3 organism groups, basically. So on the left, you'll see I've got it color coded here. Your first group is what we're calling nutrient storage. These are your base organisms. They're the ones that sequester all the nutrients into their bodies. They do all the mineralization, the micro and macro aggregation, and pull up those nutrients into their body to be released. So that's why we call them nutrient storage. So you get into that mindset of where are the nutrients going? These are the base organisms that capture those nutrients into the bodies. Then the next group is what we call nutrient cyclers. So there would be the second trophic level. They're the predators, the ones that eat the base organisms, the bacteria and the fungi. So these are these are also delineated out by aerobic. So these are your aerobic nutrient cyclers, the beneficials. So these are your flagellates, amoebas, your bacterial feeding nematodes, all your beneficial nematodes, your fungal feeders and your predatory nematodes. And then the last category is your anaerobic indicators. So we've got an anaerobic indicator in a nematode called a root feeding nematode. We've got an anaerobic indicator in a protozoa, which is called a ciliate. So they're facultative. Technically speaking, they can swing between aerobic and anaerobic, but they definitely start waking up when it starts dipping into that anaerobic zone. And then finally, the omycetes. So all of these are either facultative or obligate anaerobe. So either they don't mind some higher oxygen, or they require low oxygen. So and then the next range here, we've got the range. So this is where we're talking about those biologically complete minimums that correlate to that ecological succession we were talking about earlier. So these are the minimums in microgram per gram of each of these organism categories. And then we have our samples. So we've got bed 1. Remember, bed 1 was the one on top with the large plants, and bed 2 was the bed on the bottom with the small plants. So right away, you can see we color code this bioassay just to make it intuitive so that you can just quickly glance and see what's good and what's not good. In bed 1, we've got, we're definitely in the green, good amount of microbial populations with your base organisms, with your bacteria and your fungal populations, your beneficial soil fungi. And then we've got great nutrient cycling, really lovely populations there. And you can see right away that in the bacterial feeding nematodes, we're getting 1,200 of those guys showing up in this bed. So that's going to be a ton of nutrient cycling going on and being delivered to those plants. And they showed it in their biomass. Yeah. And in the anaerobic category, you do see a little bit of ciliates waking up there, but that's not past the threshold that we would say, okay, now you're going into really cautionary anaerobic conditions. And then you did have a little bit of omycetes showing up there as well, but we're not as concerned about this population because in soil food web speak, as microbes compete with each other, we are out competing with the number of beneficial fungi up above at that 563. We are out competing those omycetes. We wouldn't want them to climb any higher, but getting back to the nematodes, that 1,200 is really what we want to show you as we follow the path of this delivery of the nematodes. And so you can see in bed 2, we've got plenty of those bacteria, and they are higher in populations because as we look down to those nutrient cycling organisms, they are much lower. And so this is where that stratification comes into play. We're getting a ton of those organisms delivered to the first bed, but not enough organisms delivered to your second bed. Mm-hmm. So we've got some there, and they may catch up given that we've got all those bacteria, but we're not getting them delivered in the same way repeatedly, because this is something we do often on a routine basis, that they're just not showing up because it's just not being we're just not distributing it as well as we intend to do. And we got no bacterial feeding nematodes showing up. So none showing up. They all got delivered to the first bed, and we would expect that because they are heavy. They sink in water rather quickly. So that's just the math of gravity and water and an organism weight. And then one thing that we did want to point out is we do have a higher population of ciliates and omycetes. And we would wonder, okay, well, if this grower is treating everything the same, and so for all intents and purposes, we're watering the same, we're doing everything the same, what might be the difference? And the difference would be, look up here at your fungal populations, your beneficial fungal populations, and what would that have anything to do with these ciliates and omycetes, right? Well, what do fungi do? They create pore space, that macro aggregation. They need oxygen in order to breathe. That's how they survive. So if you've got low populations of fungi, of this beneficial category, they're not going to be able to swing, they're not going to be able to create enough pore space to overcome the oxygen demand of what that plant and that terrain needs in order to keep the ciliates from waking up, in order to keep it in a terrain that selects against omycetes. So who's winning here? It's the anaerobic indicators, and we just don't have enough oxygen in the system because we're not managing for that. And it's not because we don't intend to, it's just because we don't understand how just in something as simple as distributing organisms effectively can affect our growth space so profoundly.
Mm-hmm. And to take this even one step further, environment or aspects of hostility have dramatic effects on below ground microbial communities. And so this is just a really simple slide meant to explain that, this particular plant in the center is getting a more intense aspect of heat. Like a lot of times in greenhouses, if you don't have the proper diffused polyweave or polycarbonate cover, those clear films actually focus light. And if you go through with a infrared thermometer gun, you can actually measure leaf surface temperatures of the various plants. And if you were to measure the leaf surface temperature of these various plants, these two might be at or below ambient temperature. And in really extreme cases, in really bad greenhouses with terrible covers, this plant that's upset might be 10 to 12 degrees over ambient temperature. We've actually seen that a lot. In one situation that was really profound is there was random root aphids spread throughout a particular facility, and we went through with the leaf surface temperature gun. There was like 12 degrees in variation between pots that were literally within 2 to 3 feet from each other. I guess simply because of manufacturing defects of the clear film or how a non-diffuse roof actually works. And in the indoor setting, this means like maybe this plant is right next to the dehumidifier, so it's getting a tremendous amount of hot air. But when this plant is under stress, especially with cannabis, they sweat or they transpire to deal with stress. And so if you're giving a very consistent amount of water to all plants, but a given plant group is experiencing higher levels of hostility, they will use their resources of water much faster than the others. As this soil ecosystem declines in water content, the organisms will either go to sleep or they will die. If they have time, they will go into dormancy. If they don't, they just die. And over time, day after day after day, if this plant is consistently under higher levels of intensity of hostility, it will have lower soil food web populations in its root zone, despite a very consistent application of organisms week after week. And this is the evolution of what we do now. This is far, far more complex than most people think of what we do. And this is an overhead view. This is something we started having our clients do in early 2018. If you're a commercial facility in a regulated state, you get per plant yields. And so what you're looking at is 8 beds, and each one of these little colored blocks is a 4x4 space with the associated kilogram weight of flour. And so on this side of the greenhouse, there's less levels of hostility. this was by far the most hostile environment, and it had to do with the way the sun would go across this property. And this particular bed was getting, 2 to 3 hours extra direct sunlight. On top of that, when this facility filled their beds with soil, they filled up all these first, and they actually ran out of soil. And so this bed had added levels of hostility, and it also had a different soil that was significantly compromised from a nutrient quantity standpoint. And so this particular farm was struggling to get up to those over 50 pounds per 1,000 square feet. And I'll go, 2-3 hours on just this concept in a future workshop. So if you want to find out more about this, like, subscribe, stay tuned, check out our website, and we'll be releasing, this is going to be an entire episode on its own of what we do in this situation. But the basic concept is to get this facility into the appropriate 50 to 70 pounds per 1,000 square feet. All we have to do is start turning some of these yellow and orange boxes green. And if you're not a regulated facility, you should still be doing this. You can get a per plant weight. You can get one of those hanging bucket scales. And even if you do a whole plant wet weight and document that, you can still at least see where the pockets of dysfunction are in your facility. And so for this particular farm, this is where the wet wall was. And so during the hot and cold time of the year, this zone right here was getting far more hostility in the forms of extremely cold air or extremely hot air. And, like I said, we'll go into this into much greater depth in future episodes.
So what are some of the steps that you can take given everything that we've just talked about, right? So you can learn how to distribute your nematodes effectively by putting them in a simple, inexpensive, scientific squirt bottle like we showed, or any squirt bottle, really. It doesn't have to be a scientific one, but just to make sure that you mix it up, extract them into a small, manageable volume of water, and then mix them before every time that you dispense them out, because they settle so quickly. We don't advocate them putting them into any sort of reservoir unless you feel like you want to do that math and you can start to actually analyze that particular reservoir and your process in doing it, and making sure that your process will actually distribute those nematodes effectively. So measure your process. That's the next step you can take. Start measuring it, or work with somebody who does measure it and measure this biological activity. Get yourself a microscope like Scott was talking about. Do that 1-to-5 ratio of your soil, and do it like we get the samples like we suggested in your pattern of distribution, and just start doing that scan. You can see them at the 10x. One thing I would mention, if you're going to mix them up, make sure that you pull that sample out before that 10-second marker. You'll have to put your dropper all the way to the bottom and then pull it out, which you can do too. That's fine, but just making sure that you understand how to actually get those nematodes out of that sample. And so, look for them in your microscope using that 10x, like he said, you'll start at one corner and then scan across, and then move up one field of view and scan across the other direction, and just keep doing that till you've scanned the whole slide, no matter how long it takes you. And then just make sure that you're counting those nematodes. That it's always good to know what type of nematode you're looking at, but just to be able to count and verify that what we're saying in this weight and distribution is even happening, that's hands down going to help you out in a big way. So, yeah. And so we do have a growers group that we want to put out there for people that like this information, want more of this information. We try to give away a lot of information in our growers group and just support each other in this journey of quantifying what we're doing so that we're all achieving the results that we want. So we do a lot of mentoring. We do monthly Q&A webinars with our growers group. We provide a lot of educational resources on the group, lots of books, lots of research papers that we talk about, and really great people in the group that are high-level thinkers and really are actively doing this work. We also offer some discounts on labs and all kinds of things that we offer to the group as well. So it's just a great hub of a resource for people, of like-minded people that want to actually succeed at this and do well, so.
Nice.
So I think we'll open it up to questions, and thanks for showing up. We certainly appreciate our first live broadcast on our channel, and we just appreciate you guys just showing up and giving us some love.
Yeah, man.
Thanks, Smileys.
Space Cat, what up?
Yep, what's up?
That's a good name. Lord of Mercy. Lord of Mercy. That's a good one. Hype for this. We're hyped for you, Statwin.
Yep.
Oh shit, what's this? Biological complete
MILF. MILF. I love that one. I like to find. I love that. That's great. I've never heard that. I'm gonna get in trouble with that.
This is fun. You click up the little links and stuff.
Yeah.
What's up? What's up? You rock, Smiley. Right on. Well, we'll be in touch. Hit the like, hit the subscribe, do all that shit. Peace, homies.
We'll check you later.
Whoops. Sorry.
Oh, wait. Did anybody have a question?
Nope, that's it. We're moving on. They just wanted to say
all
what's
right.
up. All right, guys. Check you on the next episode.
Okay, we'll see you later.